Mujib sir, now you can start, sir.
Thank you, sir. I welcome to all delegates and presentee in this session on the behalf of Alvar Pharmacy College, Alvar, in this one-day virtual national conference in the association with IPGA. So we have selected only 5 oral presentation on the session. So now I would like to call upon
take another 1.5
Nupur Srivastava. Are you here?
So by up to 2 o'clock it will be there.
Yes, she is here.
From
Okay,
3 to 4.
Yes, sir, I'm here.
On the topic of screening of excipients for the formulation of taxophene and vivaritol self nano-emulsifying drug delivery system for the treatment of breast cancer. So
I'll make a call to you.
now I hand over the session to Ms. Nupur Srivastava.
Yes, sir, I have seen this. Yes, sir, 3 to 4. Only price announcement and validity.
How to show poster. Poster कैसे share करूँ?
Mujib sir, make
Yes, sir.
Parul Srivastava as co-host so that she can share his screen. Share her screen, sir. Make Nupur Srivastava as co-host.
Okay, sir. Okay, sir, I'm showing.
Or let me share her screen. Please wait for a while.
Okay, sir.
I think this one. Is it visible? Mujib sir, is it visible?
Not clear.
Yes, sir. This will not be clear, sir. But the name is Nupur Srivastava, and her topic is screening of excipients for the formulation of tamoxifen and resveratrol self nano-emulsifying drug delivery system for treatment of breast cancer.
It's visible.
Okay. Now she may start. Nupur, please unmute your video. Please show your video. Nupur.
Yes,
Yeah.
sir. Am I audible to everyone?
Yes,
Yeah.
yes, please.
Sir, my topic is screening of excipient for the formulation of tamoxifen and resveratrol self-nano-emulsifying drug delivery system for the treatment of breast cancer. Sir, I'm working over the tamoxifen. Tamoxifen is a gold standard drug for the treatment of breast cancer. But because of some pharmaceutical issue, there is some drawback associated with the conventional drug delivery of this drug, like the low solubility as it belongs to BCS class 2 drug, first pass metabolism as it is a substrate to PGP glycoprotein efflux pump. So because of this pump, mostly this drug get excluded out from the site of action and which lead to reduced oral bioavailability. So that's why I have combined this drug with the resveratrol, which is a natural antioxidant drug itself having the cancer properties. And but this drug is also a BCS class 2 drug. So that's why I have combined these two drugs, and resveratrol is a PGP glycoprotein blocker. So when I combine these two drugs, tamoxifen and resveratrol, then it block the PGP glycoprotein and enhance the concentration of tamoxifen at the site of action. Sir, am I audible?
Yeah, yeah.
Yes.
Carry on.
But the tamoxifen resveratrol is also a BCS class 2 drug. So that's why I have combined these two drugs in a single formulation. And for enhancing its bioavailability, I have combined these two drugs in a lipid-based formulation, that is SNEDS, self-nano-emulsifying drug delivery system. It is an isotropic mixture of oil, surfactant, and co-surfactant. It is a pre-emulsion. It is a mixture. You can call SNEDS as a pre-emulsion. So I have loaded both my drug in this formulation, and for this and for the development of this formulation, I have selected the I have selected the liquid lipid on the basis of solubility of tamoxifen and resveratrol, surfactant and co-surfactant on the basis of miscibility with the selected lipid. And with the help of pseudoternary phase diagram, I have selected the concentration and ratio of surfactant, co-surfactant, and liquid lipids. So this is the methodology. Like I have said, tamoxifen and resveratrol loaded SNEDs were prepared by novel simplified emulsification method, served with the help of exhaustive screening of oil, surfactant, co-surfactant mixture was done by using solubility studies, miscibility, emulsification potential. So I have selected the ratio of surfactant and co-surfactant ratio of lipid surfactant and co-surfactant is usually called as a S mix. So I have selected the ratio of S mix 4:1 on the basis of pseudoternary phase diagram, and liquid lipid and S mix in the ratio 1:4. And then I have loaded my both the drug in this composition and find out the droplet size, which I have found around 108 nanometer. And I have done some studies also, like stability study. I want to see the robustness of my formulation. So I have dilute my formulation 100 times with the different buffers and found the stable formulation after different dilutions also. And sir, I have used CCRD, Central Composite Design, for optimization of my formulation. So these are the plots for find out the uh, particle size. PDI, percent transmittance.
Have you completed?
Yes, sir.
Okay. Thank you, Nupur Srivastava, for the presentation. And sir, there is any question from the presentation? Naveen Gar, sir?
Sir, actually, this is the first part of my work. I'm planning for doing some cell line studies also. Like I want to find out the IC50 value of my drug also. Like what is the effect of this combination?
Both of these drugs are BCS class 2nd drug, belong to BCS
yes,
class 2nd. Yes. Tamoxifen as well as resveratrol. Okay, then self-nano-emulsifying drug delivery system you have chosen.
Yes.
Is this first time that the self-nano-emulsifying drug delivery system is prepared for these drugs in combination, or earlier this type of product has already been prepared?
No, sir. This is the first time when these two drugs are combined in the self-nano-emulsifying drug delivery system. I have found lots of paper which support this combination only on the cell line, but not any formulation I've reported for this combination.
Okay.
Okay.
So cell line studies are still remaining, I think.
Yes, sir. I'm planning for cell line studies.
Okay, good. Okay. Go ahead.
Okay.
Thank you, Nupur Srivastava. and she's, I think she's doing PhD from the Jamia
Yes, sir.
Hamdard.
Yes,
Okay, under the guidance of the Dr. Javed Ali and
and
Dr.
Dr.
Sanjay.
Sanjula Babuta is my supervisor, and Dr. Javed Ali is my co-supervisor.
Okay, okay, nice. And
Dr. Gurpreet,
thank you, thank you so much,
Yes,
ma'am.
sir.
sir, can you make
Now,
it as a full screen view? The double click on that work. you see-
Sir, this
Yeah.
is only PDF, sir. This
Yeah,
is only
PDF.
PDF.
You please double click on that. Full screen, it will be there option.
Okay.
okay.
There are export PDF and menus are there. Menu hide कर सकता है. You can do
Okay.
hide the menu.
Thank you. And now I would call Miss S. Jayakumari from the Department of Pharmacognosy, School of Pharmaceutical Sciences, Wells, Institute of the Science, Technology and Advanced Study, Palambram, Chennai, Tamil Nadu, India. Are you hearing me? Miss S. Jayakumari.
It's not there. She's not there.
I think she's not there. So I would like to call upon Mr. Ayush Garg. Ayush Garg is there? I think
He is also not there, sir.
he's also not there. Kyle Vizi is there? I think she is not. She's also not. And the fourth is Dr. Muhammad Usman Ashar.
Yes, sir.
Yeah, he.
Where?
Yes, sir. I am. Good morning, sir.
Good morning.
Sir, can I present?
Yeah. I would like to call upon Muhammad Usman Ashar, Department of Pharmaceutics, School of Pharmaceutical Education and Research, Jamia Hamdard, New Delhi.
Yes, sir.
The topic is formulation and optimization of the bromocriptine
Yes,
and
sir.
glutathione nano emulsion for the treatment of Parkinsonism disease.
Yes, sir.
I think you are also doing your PhD under the supervision of Dr. Sanjula Babuta and Javed Ali.
Yes, sir. Dr. Sanjula is my supervisor and Professor Javed Ali is my co-guide.
Okay.
Yes,
So
sir.
now I hand over to you to
Okay,
present
sir.
your oral presentation.
Okay, sir. First of all, thank you so much for providing me this opportunity to present my poster. Sir, basically, I prepared the formulation of bromocriptine and glutathione-loaded nanoemulsion, particularly for Parkinson disease. Sir, as you are aware that bromocriptine is a well-known dopamine receptor agonist and used in the treatment of Parkinson disease. Previously, also people used bromocriptine in the treatment of Parkinson disease, but this time I added glutathione, which is also have antioxidant properties for the treatment, and it also been reported that it have anti-Parkinson effect. So for that effect, we tried to develop the formulation of nanoemulsion containing both bromocriptine and glutathione to for the combined, or you can say to check whether it is synergistic effect or not. But before to start it, we choose the formulation nanoemulsion to check because nanoemulsion is a lipid-based formulation which is used to enhance the solubility of poorly water-soluble drugs. So for that purpose, we use to prepare the nanoemulsion, and we prepare the nanoemulsion through ultrasonication technique. But in this poster, I am basically emphasizing on the optimization process which I used for the selection of my formulation. And for that, I used prepare use the design expert software. Basically, I use the CCRD software in which we have to take independent variables and dependent variables. And I use the three independent variables in them. These are the oil. S-mix is basically the ratio of surfactant and co-surfactant and solution time. Whereas we choose the dependent variables are particle size, beta potential, percentage transmittance. And PDI were selected as independent variable. We applied the CCRD, and then we check the whether what are the effect of different variables, so that on that basis we can select the formulation. We generated different runs, and the software, as I showed in my poster, give 9 possible interactions, 12 possible interaction between different variables. From the results, we say that after putting the data of different variables, the optimization model suggested that all the 4 were quadratic equations except for percentage transmittance. And from the result, we can say that oil concentration has a significant effect on droplet size, followed by sonogation and S-mix. In the case of PDI, we get oil had a significant effect, same. In the case of zeta potential, finally, and also oil has a significant of transmittance. So from the result, we can say that the oil is the most significant effect in the nanoemulsion, followed by S-mix and sonogation time. So it gives the idea that how we can optimize our formulation and what dependent variable, how it affects. In the summary, sir, we can say that the droplet size of formulation increased on increasing the oil content and decreased on increase the content of surfactant due to the reduction of interference energy of observation. Also, with increase in oil concentration, there was increase in PDI, as we see in the plot, whereas increase in surfactant and sonogation time, there was decrease in PDI. Also, there was decline in negative value of zeta potential with an escalation in oil sonogation time. Percentage transmission was associated with droplet size in an inverse direction. So, sir, in this poster, I generally, I concept the optimization process, which is a very valuable parameter whenever you optimize or prepare the formulation. And so now I am also doing the further studies after the selecting the optimization like characterization and other animal study to confirm whether the both drugs have synergistic effect on Parkinsonism disease or not.
Okay. Thank you, Mr. Osama.
Thank you,
sir. There is any question from the audience side? Sir, Vishalgar sir, are you hearing me? Any question?
हाँ ओसामा मैं ये पूछना चाह रहा था कि कोई मार्केटेड प्रिपरेशन है इस कंपोजिशन में?
No, sir, not yet. Bromocriptine we got separately, and glutathione is basically is available in the form of the skin whitening agent. But as you see that where we can go through different papers, whenever anyone do for example, neurological study, they in biochemical estimation they always assess glutathione. Why? Because it has the antioxidant effect in brain, and it also exists as natural antioxidant in the brain. So for this purpose, we combine these two to check what is the effect will we get when we combine these two drugs.
Okay.
So, what are the 3 major parameters you selected for independent variables?
Sir, independent variables, I selected percentage of oil, percentage
what was the concentration range of oil you selected?
So
percentage or concentration?
1 to 2%. And I selected ethnics 9 to 13% and sonogation time in the between 40 to 60 seconds.
In -1, 0, and +1 order.
Yes.
In design expert.
Yes, sir.
Which software you selected? Which version you selected?
Sir, 11. It was 11 at that. Now it's modified, updated into 12 one, but at that time it was. They updated to 12 now.
Okay. Are you further performing annual studies?
Yes, sir. I will back to you. Because of the corona, sir, my studies are actually affected by this. So now the things are normalized. Actually, in Delhi, is still there are major scenario here. So whenever things will get normal, I will definitely do.
Okay, good. Best wishes. Thank you.
Thank you so much, sir.
Any other participant is there? No. Thank you all for your presentation, and I wish.
Hello, sir.
Yes.
Yes, sir. I'm Bhakti Mahendra Pawar from. Actually, I have submitted my abstract.
So I think we have only selected five abstract.
Okay, okay, sir. Fine. Okay, sir.
For the oral presentation, but there is only two are here. But so, are you interested to present your oral presentation?
Hello.
Yes, ma'am.
Yes, sir. I'm ready to present.
But we have no sir, Gurpreet sir.
Yes, sir.
What we can do for in that case?
So we have selected only five presentations, five abstracts for the oral presentation. What's her name? What is her name?
Miss Bhakti Mahendra Pawar, sir. I have already sent you the e-poster also.
E-poster.
Okay, I think you may be in e-poster.
Okay, okay.
This is oral presentation. Just after the completion of oral presentation, we will start e-poster presentation. So please wait for some time.
Okay, okay, sir. Okay, sir. Thank you.
We will announce your name as your turn will come.
Okay,
Okay.
sir. Okay, sir.
You
Okay,
please
sir.
stay with us. Please stay with
Okay,
us.
sir. Yes, 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 sir.
Okay.
Thank you all for your presentation, and I wish all the participants all the success. I wish all the participants to all the success. Now I hand over the session to Dr. Gurpreet Singh for the e-poster presentation.
Nothing.
No.
Thank you, sir. Now, moving ahead for e-poster presentation, I, Dr. Gurpreet Singh, Associate Professor, Alwar Pharmacy College, Alwar, would like to start this session. Thank you, Dr. Jayabalan, sir, for giving this opportunity to moderate the e-poster presentation section. And for this, first of all, I would like to call Mariette Bezu of Department of Pharmacy, Nehru College of Pharmacy, Thrissur, Kerala. Are you here? Mariette Bezu. Mariette Bezu. Please unmute yourself. Mariette Bezu, please unmute yourself for poster presentation. Mujib sir, can you please see the name of Mariette Bezu? I think he is there in the panel, but
Married Vizu, no?
yeah, he is there in the panel. Mariette Bezu, please unmute yourself. Start your video. Mariette Bezu.
Good morning, sir.
Good morning.
Please start your video, Mariette Bezu.
It is open to
We have selected 18 e-posters for the presentation, 18 abstracts for the e-poster presentation, and the first abstract of Ms. Mariette Bezu. Mariette Bezu, please start your presentation. You have 2 minutes for presenting this e-poster, and then after we will ask a few questions on that. So total 3 minutes are there. Over to you, Married Ritu. Please start and explain your process briefly in 2 minutes. Hurry up.
Good morning, sir. I'm here to present on the topic.
Married Ritu, if you are in log in into two windows, either in phone and in laptop, please mute one.
Okay, okay, sir.
Otherwise, the echo will produce.
Okay, sir. This is a questionnaire-based survey on the
No,
knowledge, attitude, and the practice of antibiotic usage among the pharmacy students. It's a cross-sectional study, and the most common drugs that are used and misused in the developing countries are antibiotics. The intense use and the misuse of antibiotics are undoubtedly the ma- The most common drugs that are used and misused in the developing countries are the antibiotics. The intense use and the misuse of the antibiotics are undoubtedly the major forces associated with the high number of resistant pathogenic and the commercial bacteria worldwide. The aim of the study was to assess the knowledge, attitude, and the practice related to the antibiotic usage and resistance. This was a cross-sectional, questionnaire study among the pharmacy students, both the B Farm and the Farm D students, and the response of the response scale ranged from strongly agree to strongly disagree, always to never, and we are using the Likert scale, and also the true or false uncertain type. The statistical evaluation was used by using the tool SPSS 20.0. In this study, the students of the pharmacy college enrolled were 443, and among them, the age group ranging from 70 to 19 were 27.8%, and 20 to 22 were 33.86%, 23 to 24 were 35.5%, and above 24 were 5.919%, and the gender-wise, 46% were males and 54% were females. And the questionnaire survey conducted based on the age and the PG or level of their graduation, and based upon the 1 second, sir. Based upon level of their graduation and age, and the PG level students were participated when the questionnaire survey conducted, and the knowledge, attitude, and the practice were evaluated. The different batches only produce some significant observation. This may be due to the lack of educational awareness. The students belong to the medical and healthcare sector should have well knowledge about the rational antibiotic and prescribing and development of the resistance for the welfare of the general masses. Actually, we have found that only the UG students and they are well in the phase of knowledge, but the practice level of the students were poor. And it is important to improve their knowledge in the antibiotic prescription. It will help for the preventing the resistance of the bacteria and will improve the welfare of the general masses. Thank you, sir.
good, Married Ritu. Anyone in the panelists, you may ask question. Dr. Mujibur Rahman, Dr. Rajesh Sharma.
Yes, one question is there. As being a pharmacist, how we can stop the resistance of the antibiotics?
So, we can control. Hello, sir. Can you?
Yeah. You please continue.
We can control by preventing the overprescription and polypharmacy, and and improve the rational use of antibiotics. Hello.
Yeah.
So there is any suggestion?
The irrational use arises from the various economic factors. The irrational use of antibiotics is arise from various economic factors and healthcare policies concerning about the medical insurance, the lack of physicians, or the concerns pertaining the long-term resistance and the effect versus treating current symptoms and the marketing of the pharmaceutic and antibiotic sale without prescribing in but the prescription in some countries. This may be the factors that may causes the resistance of the bacteria.
Okay. Thank you, Ashida.
Thank you, Mario. Next presenter, please be ready. Mr. Ayush Garg. Mario, now you can stop your video. We will announce the results later in the validatory ceremony. All the best. Next presenter, Mr. Raghavendra Banjimala. Mr. Raghavendra Banjimala. Is Mr. Raghavendra Banjimala is there? Mr. Raghavendra Banjimala. Remaining participants, please mute your audio, sorry. Remaining participants, please mute your audio. Is Mr. Raghavendra Banjimala is there?
I think he is absent.
Okay. Moving upon next. Miss Debolina Saha. Miss Debolina Saha.
He is there.
She is there. Debolina,
Yes,
please start your video and unmute. Yeah, you are unmuted. You may start your presentation on role of 3D and 4D printing in pharmaceutical industries in 21st century. Please briefly describe your e-poster.
Hello, sir. Am I audible?
Yeah, you are audible. Please.
Okay, sir. Sir, today my presentation on the role of 3D and the 4D printing in pharmaceutical industry. First of all, myself Debolina Shah, B.Pharm final year student of Guru Nanak Institute of Pharmaceutical Science and Technology, Kolkata. Because sir, the nowadays, the 21st century, the use of the 3D and the 4D printing technologies are increased by day in the globally pharmaceutical industry. 3D printing plays an important role in the customized medication and the pharmaceutical R&D department as well as the healthcare system also. The 4D printing is the new innovation which is invented in 2015 and uh, and first it is the published in 2030 in a conference also. Then what is the basis of the 3D printing? The 3D printing is called the additive manufacturing process also. It can be said it is the layer printing because this is also known as the stereolithography, which is based on the layer. First of all, in the FDA approves the 3D printing drugs are the spritam, which is the generic name is the levetiracetam. The company Apprecian, the USA based company, in 2015 August first marketed this drug. It is an anti-epileptic drug. The 3D printing technology is based on the process. First of all, in software, through software, the drug designing is done. Then then with the stereolithography process, the medication will be done. There is a several application for the 3D printing in the pharmaceutical field, like uh, the first of all the personalized medicine and the customization medicine, as well as the different body parts for the for human, like the implants also. So in this presentation, I mentioned the 4D printing also. The 4D printing is first invented by the hello.
Yeah, you are audible. Please
Huh.
continue.
The 4D printing first invented in.
Debolina, you are audible. Continue.
Have you completed your presentation?
Hello.
You are audible to us, Debolina.
Yes, sir. Besides this 4D printing developed in the collaboration with the MIT company, MIT company, with which is a self-assembly lab, and it is declared by the CEO of this company. In case if we ever see, if we see the 4D printing technologies, it is a totally new innovative things in our world, and especially in the pharmaceutical industry. In health industry, it is started to develop different models like the 3D printings of spinal cord or the jaws, etc. In my poster, I also explain it through the diagram also, and the graphical presentation also, how it is impact through all over the world. If we believe this 3D printing technology and the 4D printing technology, it can be seen that the 4D printing is a 3D printer with a multi-smart material, and which is more upgraded than the 3D printing. 3D, totally is the 4D printing is a novel concept, and which is independently alter their morphology before the 3D printing. And if we see the future aspect of the of the 3D printing and the 4D printing technology, we can see that it is in 2020, where it is the approximately $6.21, and it will be growth with 21 point and more dollar by 2021.
Okay,
In my
thank you,
conclusion,
Debolina. Yeah, please conclude, please.
yes, ma'am. Sorry, yes, sir. In my conclusion, I want to say that although the practical formulation of the such novel dosage forms and delivery system is complicated and difficult, the scientists are the certain that the modern pharmaceutical industry is seeing a turning point, and that the 3D printing is set to revolution the way we look at the medicine and the medication. No need to surprise if you see the 3D printer is in every doctor's office or hospital in the very near future. The 4D printing technology is expected to significantly in this way to increase the efficient the manufacturing process in pharmaceutical industry. Thank you, sir.
Thank you, Debolina. May we have the questions from the panelists?
Yes, sir.
Any panelists want to ask question?
Devolina, do you know about the stereolithography, which is used in 3D printing?
Can you repeat this question again?
Stereolithography,
Yes, sir.
which is used in 3D.
Yes, sir. Stereolithography is nothing but it is a layering technique, which is a UV procedure technique. If we consider how does the stereolithography is work, then you can see the stereolithography is a laser based technology that uses the UV sensitive liquid resin. The UV based liquid laser beam scans the surface of the resin, and it work under the it was under the giving input of the software. Here we can just like in 3D printing, we use the software like the area the repeated heat and the Cura also.
So which technique is more beneficial, 3D or 4D printing?
Sir, first of all, the technique is beneficial for both. In case of if we see which is the more beneficial, then we also can say the 3D printing is more beneficial, because 4D printing is totally new innovative thing, and it is the it does technology depend on the 4-dimensional technique. And there is a totally different from the soft the main principle is same, but the software and the technology of the processing is totally different from the 3D printing.
Okay, Debolina. Thank you for your justification. All the best. Now I would like to call Nimisha, RN Nimisha of Department of Pharmacy Practice, Nehru College of Pharmacy, Thrissur, Kerala. RN Nimisha is here.
She is not here.
Not here, sir.
Yeah.
Okay. Moving on. Next. Anna Dhill baby. Is Anna Dhill baby is here?
She is there.
Yeah, Anna Dhill. Please start your video. Yeah, you are audible. Please start your video. Just start your video. Yeah.
Good afternoon, everyone.
Good afternoon. You please be seated.
Is it visible?
Yeah, you are visible. I think your presentation is on clinico-etiological evaluation and management of electrolyte abnormalities in hospital patients. Is
Yes,
that
sir.
right?
Yes, sir.
Okay, please start. You have 2 minutes to briefly explain this poster, and 1 minute will be given to you for the answers.
Okay, sir. So in our study, we are finding out the clinical and etiological factors which are responsible for the electrolyte abnormalities in the hospitalized patient. So in the study, we find out what are the different types of electrolyte abnormalities like hyponatremia, hyperkalemia, and hypokalemia, and then the abnormalities of calcium and chloride. So our study has a total population of 175 patients, and all are aged above 18 years and above are included in the study. And we conducted the study for a time for 6 months. And then, according to our result, hyponatremia is the most common electrolyte abnormality found, followed by the hypokalemia. And then the main symptoms of the patients was the weaknesses and vomiting. And then when we are considering the pre-existing diseases, most of the patients with hypertension and diabetes mellitus had electrolyte abnormalities, and the hypo and hypercalcemia were effectively, and we also consider the management of all these electrolyte abnormalities. And in case of hyponatremia, it is mainly treated with the 3% NaCl, and also in case of oral treatment, they have given tolvaptan drug, and in case of hypokalemia, they have given drugs like salbutamol plus insulin combination, and also the injection KCl for hypokalemia. And then when we are considering the ratios, our male patients are more affected than the female patients. And then in case of hypochloremia, it is more reported in the patients um, with chronic lung diseases. And according to our study, we can say that the electrolyte abnormalities are very common in almost all the hospitalized patients, and we have to what we need is regular monitoring of the electrolyte, and we have to stop any offending drug if anything is there.
Okay, have you completed?
Yes, sir.
Okay, any question from the panelists? Rajesh sir? Dr. Rajesh Sharma, any question? Gaurav Kumar, Gaurav sir, Gaurav Soni sir, any question?
आपने स्टडी जो पेशेंट पे आपने की है 175 पेशेंट एज 18 पे, तो कंक्लूड क्या निकला कि आपने जो एडमिटेड पेशेंट पे आपने जो सर्वे आपने किया इलेक्ट्रोलाइट इम्बैलेंस का, तो रिजल्ट क्या निकल के आया आपका, क्या था?
Actually, we took 175 patient case sheets, and almost all of them had these electrolyte abnormalities.
Yes.
Okay, thank you.
Okay.
Yes, sir. You please continue, sir.
Can you tell us the some drugs which is shows the side effect as a hypokalemia?
Hypokalemia. So in case of diuretics, in some cases they have given fluoride, diuretics.
Okay.
AC inhibitors.
Okay. Thank you, Anandin. All the best. Now moving up on our next participant, MP Narmada. Narmada. Narmada.
MP Narmada.
MP Narmada. Is MP Narmada is there?
Sir,
Yeah.
sir, it's not MD Narmada, it's presented by Lina Chandichu.
Sorry. Yeah, you are right. You are right. One second. Linus and Desu. Is this?
Which, sir? Yes, sir.
Okay. Okay, Linus and Desu, your present topic of presentation is drug utilization of antihypertensive drug in hypertensive patients in a tertiary care hospital. Is it? Okay, Lina, you please start your
Yes, sir.
presentation. You please start briefly explain your poster.
Yes, sir. So, good afternoon. So, my topic is on drug utilization of antihypertensive drug in hypertensive patient in tertiary care hospital. So, as hypertension is a leading common disease that causes very much death recently, so we have thought of doing a study to find out whether the drugs that is given to the patient is in an efficient way. So in our study, we mainly focused on to find out what is the mostly prescribed drug in our hospital, and we were able to find that calcium channel blocker was mostly prescribed, and among them, amlodipine was mainly encountered to the patient. And we also find out what is the main social risk factor for hypertension, causing hypertension in individuals. And we were able to find that alcohol was a leading cause at 25% of patients were intaking alcohol and they had hypertension, and smokers also had hypertension. And we were also able to find the duration of hypertension was almost 10 years. Mostly the patient had a duration of 10 years vastly. And we found that in our hospital monotherapy was mostly prescribed in patients than the combination therapy, that is about 52 percentage of monotherapy was administered to the patient. And by this study we also find out whether our hospital is following which guideline. We found out that our hospital is following JANC 8 guideline. And we found and in our study, almost all patients with a comorbid condition and with an only a hypertensive patient also followed JANC 8 guideline. And the BP goals were also achieved in about 82% of patients achieved BP goals by the treatment that is given in our hospital. But in CKD patient, a mild variation was seen. The achievement of BP goals was decreased even after the treatment was continued. So this was our results after our study.
Okay, thank you, Linus. Any question from the panelists to be asked from Linus?
Leaner,
Yes, sir.
anti-hypertensive drug एक फिक्स टाइम डोज दी जाती है. फिक्स टाइम, व्हाट इज द मीनिंग फिक्स टाइम क्यों दी जाती है?
Yes. Fixed?
Fixed time.
Sorry, sir, once more. Fixed.
Why fixed time doses are required for antihypertensive drugs? Have you heard the term fixed time?
Okay, Lina.
I think she is disconnected or
I'm
is
not
connection
familiar with that
poor.
term, sir. Hello?
Tell us about the JNC 8 guidelines.
So JANC 8 guidelines mainly focuses on patients with that.
First, you tell us full form of JNC.
Hello?
Yeah, you are audible.
First, you have told us the full form of JNC, and then tell us the JNC 8 guidelines.
What is the full form of JNC, Lina? Sir is asking what is the full form of JNC? Are you hearing?
Hello.
Yeah, you are audible to us.
JNC 8 guidelines joint national committee, and the guideline mainly focuses on based upon the age and the therapy that is given to the patient.
Okay.
Hello.
Hello.
Yeah. Can you please describe the various stages of hypertension?
Stage 1 hypertension, there are mainly stage 1, stage 2, stage 1.
Echo sound is produced because you have connected from two different systems. Echo is producing, that's why. Please mute one from one system.
Hello. Hello.
Okay. Lena, please tell. Please
Yes,
speak.
sir.
Yeah, you are audible.
Mainly hypertension are divided into 4 stages. That is normal, pre-hypertension, stage 1, and stage 2 hypertension. So,
Okay, thank you. Thank you, Lena.
yes, sir. Thank you.
Moving on, upon next is Ankit Mazi is there?
Yes, sir.
Yeah. Ankit Mazi, please start your presentation on implementation of artificial intelligence in clinical trial. You have selected this topic.
Okay, sir.
Okay.
Good afternoon, sir. I'm Ankit Maji from Guru Nanak Institute of Pharmaceutical Science and Technology. Today, my topic is implementation of artificial intelligence in clinical trials. So, clinical trial, it has been trending nowadays. So, the main target of the sponsor remains in increasing the accuracy of the clinical trial and decreasing the cost of the clinical trial. And the best way is using an artificial intelligence. So what is an artificial intelligence? It is a mimicking of human intelligence with the help of a software or any computer technology. Now artificial intelligence has got a vast role in healthcare system, starting from diagnosis to continuous measurement of various live processes and generation of 3D generation of 3D printed organs and all. Now, what is the role of artificial intelligence and clinical trials? So, first one is using it to record the adverse event information, the MHR and then EHR data, generation of preclinical data sets, DNA database. We can create the DNA banks, the gene banks. We can collect the real world data and evidence. We have the mobile application like PVPI, ADR reporting apps. We can use computational analysis for data generation by using algorithms such as neural networkings. We can do machine learning. We can create personalized medicine, which is the future of medicine. So generation of next generation antibiotics, we can create the novel digital endpoints like the health tracking watches. We can create the health prediction apps, which can assess your health status with the help of taking the data from the patient. So coming to the conclusion, we can conduct home-based clinical trial. The cost will be much reduced, and the accuracy of the clinical trial will also increase. But there are some problems regarding this technique. So first of all, we have to train the personnel regarding this, and as lots of data will be generated, so data security is a major problem for this artificial intelligence. And the initial investment for setting up this is quite high for the sponsor. Thank you.
Thank you, Ankit. Do we have questions from the panelists?
Yes, Ankit. Can you differentiate artificial intelligence clinical versus human clinical trials? how you can differentiate in between?
Yes, sir. Sir, artificial intelligence can be used in the ex vivo technique. We have the ex vivo technique, such as we can perform experiments in rabbit eye, then ileum, with the help of various softwares available. And again, in clinical trial, we can directly record the patient data and send it to the server so that it becomes easily available to the physicians and the sponsorer.
Okay, okay. Thank you, Ankit. Moving on upon our next presenter, Ms. Farooq Sood. Farooq Sood, please unmute and start your video. Is Farooq Sood is here or not? Farooq.
Is there?
Yeah, she is there, but I think she is not listening to us. Farooq, please unmute yourself. Okay. Moving towards our next topic, next presenter, Dr. M.P. Narmadha. Dr. M.P. Narmadha. Is Dr.
Good
M.P.
afternoon, sir.
Narmadha? Yeah, yeah.
Sorry, sir, it's not Dr. MP Narda, it's our guard. It's myself, Ashisha B.
Okay, okay, okay,
Okay.
Sir, am I
Ashish,
audible?
Ashisha. Your name is
Yes,
Ashisha.
sir.
Okay. Yeah, you are audible, Ashisha. Please,
Okay.
I think your topic is assessment of cost of illness and bone disorders of pre-dialysis and dialysis patients in a tertiary care hospital.
Yes, sir.
I think you all are from the same department, na? Nehru College of
Yes, sir.
Pharmacy.
Department of Pharmacy Practice from Nehru College
Okay.
of Pharmacy.
Okay, good contribution from you all.
Thank you so much,
This is,
sir.
yeah, please start your presentation.
Okay, sir. So myself, Ashisha B from Nehru College of Pharmacy, presenting our 5th year project that my topic is assessment of cost of illness and bone disorders of pre-dialysis and dialysis patients in a tertiary care hospital. Cost of illness study is one of the pharmacoeconomic evaluation method to estimate the overall cost for a particular disease in a defined population. And the CKD mineral bone disorder refers to the disturbances of mineral and bone metabolism which occur in CKD patient, which is associated with bone loss, fractures, cardiovascular disease, decreased quality of life, and increased mortality. The aim of our study is to assess the cost of illness and bone disorders of pre-dialysis and dialysis patients. A prospective observational study was conducted over a period of 6 months at nephrology department of our hospital, PK Das Institute of Medical Science in Palakkad, Kerala. Recruitment of patients were based on the selection criteria, that is the patient who are on dialysis, who are on treatment with vitamin K27 tablet, and those who are on pre-dialysis group, that is stage 3 to 5. Bone mineral assessment was done by screening biochemical parameters like serum calcium, phosphorus, vitamin D, and intact PTH. The data pertaining to a cost of illness, including direct and non-medical costs, were collected. Patients with low bone turnover were treated with vitamin K27 tablet, and the biochemical parameters were screened after 6 months. Our study shows significant strong correlation between the length of hospital stay and the total direct cost. The ABD, that is adynamic bone disease patients, after treating with vitamin K27, showed a significant increase in the IPTH values. So, calcium, phosphorus, vitamin D, and IPTH were the evident markers of CKD MBD in study patients. Vitamin K27 therapy was very well effective in treating adynamic bone disease patients group who the intact PTH level was less than 150 picogram per ml. So this was a safe nutritional strategy to be integrated into the pharmaceutical care in treatment of patients with adynamic bone disease in dialysis group. Thank you.
Okay, Ashisha. May we have the questions from the panelists? Gaurav sir. Yeah,
No, sir.
Gaurav sir.
No, sir.
Okay.
Can
Sharma
you
sir. Yeah, Mujib sir. You please
tell
ask.
us what is the tertiary care in hospitals? Can you explain?
The tertiary care hospitals, mainly the large one hospitals like the 750 or more than 750 bedded hospital who have super specialty care, including nephrology, super specialty neurology, and surgery related. And it's a referral center with level of healthcare that is obtained with the specialist in overall in all diseases.
What are ABD?
Sorry, sir.
What are ABD?
It's adenamic bone diseases. It's like a low bone turnover disease, mainly occurring in patients with CKD due to the imbalance in the homeostasis of calcium and vitamin D. So it's a variety of renal osteodystrophy characterized by low osteoid formation and is generally called adenamic bone disease with low levels of intact parathyroid hormone.
Okay. Okay, Ashisha. All the best. Thank you.
Thank you, sir. Thank you so much.
Moving on to our next presenter, Aishwarya Sunil. With Aishwarya Sunil is there?
Yes, sir.
Okay, Aishwarya, please start your video first and then start your presentation. Yeah.
Okay,
Aishwarya,
sir.
yeah. Study on risk factors and drug utilization pattern in stroke patients in a tertiary care hospital.
Yes, sir. Good
Okay.
morning, everyone. Myself, Aishwarya Sunil from Nehru College of Pharmacy. I'm a fifth-year PharmD student presenting our project. The stroke continues to be one of the leading causes of morbidity and mortality. The risk factors of stroke include diabetes mellitus, hypertension, dyslipidemia, smoking, alcohol consumption, etc. This study was conducted to obtain the prescribing pattern of drug in stroke patients, thereby analyzing the extent of its rationality. The aim of the study was to study the risk factors of stroke and the drug utilization pattern in the stroke patient in order to scrutinize the extent of rationality in the drug prescription and to bring out the pharmacist's intervention. The study was a prospective observational study of 6 months, which was carried out in Pikeda's Institute of Medical Sciences, Vaniyakulam, and the data were collected from the patients admitted to the Department of Neurology, satisfying the inclusion criteria from the medical records with the help of a standardized data collection form. The patient demographics with risk factors, associated comorbidities, and the prescription pattern of the drug were also recorded. In our study, among 101 subjects, the incidence of stroke was predominant in males with 53.46%. Most of them were seen in the age group of 61 to 70 years, and among the risk factors, the hypertension was found to be the most prominent one with 73.2%, and the least seen was old CVA with 9.9%. On analyzing the prescription pattern, antihyperlipidemics were prescribed to all the patients, and the average number of drugs per prescription was found to be 9.79, and the total drugs were 989. The total adherence of the treatment with the standard AHA or ASA treatment guidelines was found to be 94.2%. Our present study helps to evaluate the risk factors of stroke and detect the polypharmacy present in the treatment along with the extent of rationality. The treatment pattern reflects the maximum rationality with the guidelines.
Okay, okay, Aishwarya. Any questions from the panelists?
Hello.
Yeah, Gaurav, sir. Yeah,
Tell me one thing that how can smoking causes stroke?
Smoking, which includes nicotine, cigarettes, and all other tobacco products contain nicotine in our blood, which can induce and decrease the metabolism of all other drugs and can increase the production of acetaldehyde, which in turn increases the more risk factors for stroke patients.
Surya, can you tell us why stroke is prominent in men as compared to the
Hello, sir.
why stroke is prominent in men as compared to the women?
In females, they have a high estrogen effect, which have a protective effect for women. So that in males, there is less estrogen and they are more prone as they are not protected for stroke.
Okay, this is the reason.
Yes, sir. Estrogen have a protective effect on women for stroke.
Okay, Aishwarya.
Hello.
All the best. Yes.
Sir, one ask one question.
Yeah, Naveen sir. Yeah,
Sir,
sure, sir. Sure. Please ask, sir.
can you tell me the
Naveen.
symptoms of stroke? What is a stroke? How can I identify the stroke?
Stroke can be defined as a
Yes.
rapidly diagnosing a loss of cerebral function, and
Yes.
the symptoms can be slurring of speech,
Okay.
paralyzing of limbs of upper limbs and lower limbs, and they can be tremor, there will be slurring speech and
Okay.
Okay, Aishwarya. Uh, Thank you for the presentation.
Thank you.
Moving upon our next presenter, that is, I think, Hema Malini B. Is there Hema Malini?
I think she's not here.
Is Hema Malini B is there or somewhere busy in film industry?
No, she's not here.
Okay. Moving upon our next, that is, who is the presenter here? No underline and no arrow mark. Bhakti and Pawar.
Yes, sir. Yes, sir. I'm here.
Okay, Bhakti, Pawar, you please first start your video. Yeah.
I'm visible, sir.
Yeah, you are visible and
Yes. Good
audible
afternoon,
also.
sir. Good afternoon.
Good afternoon.
My topic is applying the simultaneous equation or Virorod's method for concurrent determination of etodolac and ibuprofen by the UV spectrophotometer in drug solution and marketed dosage form. Basically, sir, we are all are aware about the UV spectrophotometer. We use this for quantitative estimation of the drugs, and we all are also aware about the combination of the drugs and the synergistic effect offered by them. So here, the objective of my research work is to develop and apply the simultaneous equation or Virod's method for concurrent determination of the marketed dosage form. Here I have used etodolac and the marketed dosage form is etofree 400 mg and ibuprofen ibugesic 200. As the marketed combined dosage form is not available for this drug, I have used the drug solution by making the powder of both the drugs, and the results I have validated according to the ICH guideline Q2R1. Then, sir, simultaneous equation method is actually applied to estimate the drug combination that contain the double or triple APIs in combination. If our sample contains the two solution or two absorbing drug, each of which absorbs at the lambda max of other, it may be possible to determine both the drugs by the technique of simultaneous equation method. As if we want to estimate the drug concentration by simultaneous method, there are n number of methods by which we can estimate, like simultaneous equation method, isoabsorptive method or isobastic method, then Q absorption ratio method and only absorption ratio method. Here I have used a simultaneous equation method as because it is very simple and precise method. By keeping the values of the absorbances and absorptivities in the equations, we can finally get the concentrations of individual drug. And see, the simultaneous equation method, what I have done is I have prepared the working solutions of both the drugs and scanned into the UV range. I have found the wavelength lambda max of both the drug 278 nanometer for etodolac and 264 nanometer for ibuprofen. And by calculating the absorptivity values putting the values into the cx and cy formula, I got the concentration of individual drug. And by cx and cy, by using that values, I have back calculated the percent drug content release of the drug there, where I got percent drug content 98.8 to 101.5% percent drug content for the etodolac. As I have prepared the concentrations from 1.85 to 29.6 for marketed tablet of etodolac. Initially, I perform the calibration curves by using the pure drug, and then I have applied this method to the marketed tablet dosage forms. And for ibuprofen, again, I have got 100% drug contained I have observed that whatever the absorptivity values I got into the pure calibration curves, it obeys the Beer-Lambert's law because the absorptivity values of all the drug concentrations are nearby. Here I am concluding that whatever the method I have developed, simultaneous equation method, is simple, precise and accurate. And the statistical analysis proved that the method was repeatable and selective for the simultaneous estimation of etodolac and ibuprofen in pure and pharmaceutical dosage form without any interference of the excipients, whichever there is in marketed dosage form. Hence, this new simple method can be used routinely for the estimation of these drugs by using simultaneous equation method. Thank you, sir.
Okay, Bhakti Pawar, you have very well explained. Is there any question from the panelists to be asked by Bhakti Pawar?
Okay, can you tell us which solvent you have used during the UV spectrum?
Phosphate buffer 6.8, sir. pH 6.8 phosphate buffer for both the drug.
Okay, okay. So how we can differentiate the precision and accuracy?
Precision is, sir, according to ICH, accuracy is true value near to the closeness towards a true value, and precision is we have to perform like intraday, interday, 1, 2, 3, and by calculating the percent recovery studies. By using the formula, we'll get precision values for the data.
Okay.
What is ultraviolet spectrum method?
Hello, sir. I'm unable to listen.
What is the ultraviolet spectrometry method? Define.
Sir, can you please tell again?
What is ultraviolet spectrometry method? Define.
Ultra UV spectrophotometric method, whatever I have performed here. Okay. First, I made the concentration range for the pure drugs, and then I scanned for the lambda max, each drug. Then, by scanning, I got individual lambda max. Again, I have used for the same method for marketed dosage form. Before that, for pure drug, I have calibrated by using these drug calibration curves. I got the linearity 0.997 for ibuprofen and 0.992 for etodolac. So the same method I have applied for a marketed dosage form. I formed the powder and then I made the concentration stock solution. From that I made serial dilutions and then I checked for the absorbance. From that values I have calculated the Cx and Cy. And in simultaneous equation method there is a proper formula for Cx for calculating the X amount of the drug and Cy for calculating the Y amount of drug of others drug, which is second drug, and Cx is one drug, ibuprofen, and another one is etodolac. By putting the values like cross values, cross absorptivity values, I got some amount for each and every absorbance, Cx and Cy. From that, I calculated the percent drug content, sir.
Hello.
Okay, Bhakti Pawar. Thank
Hello.
you.
Okay, sir.
Hello.
Thank you.
Yes, sir. Yes, Gaurav, sir. Do you want to ask?
Yeah, sir. Is there any combination product of etodolac and ibuprofen in market?
No, sir, not yet.
Not yet.
Actually, yes, sir. That's why I try to see what the two NSAIDs, as these both the drugs belongs to the NSAIDs categories. The limit for the NSAIDs categories in formulation is only maximum 2. So a combination of these two drugs is not available yet. But with ibuprofen, another drugs are available, not ibuprofen, etodolac. So I try to take individual drug combination and make the drug formulation, drug solution.
Okay.
Showing
Why
synergistic effect.
you have chosen phosphate buffer pH 6.8 for making solution, stock solution? Phosphate buffer pH 6.8. Okay, I think she disconnected.
Hello, sir.
Yeah, Bhakti.
Yes, sir.
Why you have chosen a phosphate buffer pH 6.8 for making solutions, chalk solutions?
Sir, as I have tried for methanol also, as it is organic solvent, but phosphate buffer, it mimics the physiological buffer in our human body. So it is feasible for if someone tries for going for marketed formulation to prepare this formulation, then
This,
phosphate buffer
I
is.
think this pH is suitable for the salivary pH. If you are mimicking the salivary pH,
No,
then
sir,
it
it
is
is.
suitable. Then it
No,
is suitable.
yes, yes. Sir, it is intestinal pH
Simulated
phosphate.
intestinal pH is not 6.8 exactly, but you can perform. Yeah.
Bhakti, why should you choose Etodolac or ibuprofen? There are many drugs available in the market for this category.
Sir, yes, ibuprofen with another drug is possible. Means the marketed formulations are there available, but not for this ibuprofen etodolac. But I have to check whether these both the drugs are showing any. Is there any interaction? I will do, sir, further studies for these combination just to check the is there any compatibility, incompatibility, or synergistic effect between them. I will go forward. I will do this study, sir. This is, sir, initial work of my study.
ओके क्या आपका जो कंपोजिशन है इटोडोलैक आइब्रोफेन कंप्लीटली क्योर द अर्थराइटिस और
Yes, sir. I didn't do the clinical means preclinical studies, but it is possible if it is showing synergistic effect, then we can go because as the limit for this NSAIDs is maximum 2, as we have seen ibuprofen, paracetamol tablets are also available. So I will try for ibuprofen, etodolac also if I get chance.
ओके भक्ति बेस्ट ऑफ लक अगर
Yes.
ये क्योर कंप्लीटली क्योर करता है अर्थराइटिस को तो
Yes.
बहुत बड़ा अचीवमेंट है
Yes.
ओके
Yes, sir. I have just developed the method for UV spectrophotometer just to study the simultaneous estimation of these drugs.
ओके थैंक यू
Okay, thank you, sir.
Thank you, Bhakti Rama.
Thank you, sir.
Yeah. Moving upon our next presenter, D. Akila Devi. Is D. Akila Devi is here? D. Akila Devi. Any from the presenter? Any author from this panel? Nobody is here. Next, shall we move on to the next topic? M. P. Narmada. Is Narmada is here or Gayatri? Yeah, Gayatri.
Am I
Yeah,
audible?
please. Yeah, you are audible.
So
Please.
myself Gayatri, MP Narmada is my guide.
Okay, okay. Please start your video, Gayatri, first.
Okay, sir. So am I visible?
Yeah, you are visible and audible. Please start.
Sir, can I ask you something? Sir, you had sent a template to prepare the poster. So I have prepared a poster. So will I be able to share it over here?
Ma'am, due to some technical problems, we can't share at this moment.
Okay. Then I'll proceed with my presentation.
Yeah, please.
Gurpreet, if she has the poster, let her present the poster.
Sir, we can't make her. You can make her, sir, only co-host, so that she
You
can
have the
share.
poster, Gayatri?
Sir?
You have the poster with you?
Yes, sir. Yes, sir. I have the poster.
You please raise your hand. Raise the hand.
Okay. How do I do that? Okay.
Yeah.
Okay. So can I share my screen now?
Now you can do it.
Can I do it, sir?
Yeah.
Okay. Just a second, sir. Sir, is it visible?
Not yet.
Not yet.
Sir, is it visible now?
Yeah, it's coming now. Yeah.
So can I start my presentation?
Yeah.
Okay, so very good afternoon to one and all. Myself Gayatri. I'm here to present my project, the study of mortality risk assessment in intensive care unit of a tertiary care hospital. So this study, my study was actually based on intensive care unit. It's just a part of my project. My project also includes the other part. So it's a part of my project. So moving on with my presentation. Now, intensive care unit provides the critical care and life support for the critically ill and injured patient. So my study aims to identify the different scoring systems, aims to study the different scoring systems that are used in an intensive care unit and to identify which scoring system will be sensitive and specific in the intensive care unit of my study center. So with that, I moved on. I had a total of 215 patients for my study. So for the group one, and it was divided into two groups, the group one survivors and group two non-survivors. So this group 1 survivors included 200 patients and group 2 included non-survivors, that is 15 patients. So here the group 1, so the group 2 non-survivors, the 15 non-survivors are those who died due to the critical illnesses. That is not, these death patients do not include those who were died of a suicide or any other reasons other than a clinical case. So further, we collected the institutional review board gave us the permission to collect the data of the death patients also. And further, we prepared a data collection form and moved on with the data collection and many laboratory values and everything was collected. And then Apache 2 and SAPS 2. Apache 2 and SAPS 2 are the two scoring system. These are not the only two scoring system. There are several other scoring systems. So we selected these two scoring systems mainly because the doctor who was in charge of intensive care unit asked us to select these scores to identify which one was the most sensitive and specific score over here in our intensive care unit. And further, using the SPSS software, we carried out the statistical analysis. So moving on with the results. So a total, as I said before, we had 215 cases, 200 patients in the group 1 and 15 cases in group 2. And further, I carried out the statistical analysis to determine if there is a significant difference in age between the two groups, and it was found that there was no significant difference. And also there was 62 in total in 215 cases, 62.3% were males and 37.7% were females. And further, on the basis of age-based distribution, we found that a greater percentage of patients were in the age groups 59 to 80, 59 to 80, that age groups were mostly observed. And further, as I said before, the laboratory parameters were collected, and we tried to identify which laboratory parameters were significantly different in the either of the groups. And for that, man-Whitney U test was performed, and it was found that the following parameters, that is the sodium, hemoglobin, RBC, PCV, MCHC, polymorphs, lymphocytes, eosinophil, ESR, serum creatinine, bilirubin, serum globulin, and serum albumin were found to be significantly different in either of these groups. So further, we moved on with the calculation of the scores. Now, the very characteristic of these scores is that the score could be used only for those laboratory parameters which is obtained in the first 24 hours admission of the patient. So the Apache 2 and SAPS 2, these two scores were calculated on the basis of the different laboratory parameter inputs that is required for the either of the scores. And then we calculated the scores for all the patients in both group 1 and group 2. And we identified that there was significant difference in these mean scores of either of these groups. And the mean values were more for the group 2, that is non-survivors. Now, the Apache 2 and SAPS 2, we used an application called as MDCalc application for the calculation of the scores. And further, moving on to these ROC curve analysis is what we use to identify the sensitivity and specificity. And with the input, using the SPSS software, with the input of data, I calculated the sensitivity and specificity, and it was found that the AUROC, that is the area under the ROC curve, was more for Apache 2 compared to the SAPS 2. So both had a greater than 0.5, but yet a greater point difference was observed for Apache 2. So we can say that a greater sensitivity and specificity was there for the Apache 2. Further, to determine whether the scores are calibrated or not, we performed the Hosmer-Lemaitre goodness of fit test, just like a chi-square test, and it was found that the p-value was greater for Apache 2, which means it had more calibration for identifying the risk of mortality compared to SAPS 2. So with these tests, with this statistical test, we confirmed that in our study center, Apache 2 was more discriminative and calibrated. So using this Apache 2 score, as I said before, in case of Apache 2, there are only 12 parameters. There are no other laboratory parameters included. So I use the parameters that are not included in the Apache 2 to identify which one could be an independent predictors of risk of mortality. So I performed a univariate binomial logistic regression, and with that I found that ESR, hemoglobin, serum creatinine, serum globulin, albumin, lymphocytes could be an independent predictors of risk of mortality. And further, I developed two models, that is one wherein we obtain only the lymphocytes, ESR and hemoglobin, and the another model wherein we would get also albumin, globulin and serum creatinine data in the first 24 hours. So in the first model, those are the patients in whom we obtained only lymphocytes, ESR and hemoglobin in the first 24 hours. And it was found that ESR could be an independent, with the binomial logistic regression, we found that ESR could be an independent predictors of risk of mortality in those patients. Whereas if we are to get additional laboratory parameter data such as albumin, globulin and serum creatinine, we can say that globulin and serum creatinine could be an independent predictors of risk of mortality as well as serum creatinine. So with this, I can conclude that the Apache 2 is most sensitive and specific score in our intensive care unit settings, and ESR and globulin are the independent, as well as the serum creatinine are the independent predictors of risk of mortality. And this three parameters could be further used by the other researchers, or if I'm going to do the further research, this parameters could be used to enhance the sensitivity and specificity of that particular scoring system. That's it. Thank you.
Thank you, Gayatri. Any questions from the panelists? Gayatri, what is AUC ROC?
I'm sorry, sir, I can't hear you. Sir, can you repeat the question?
What is AUC ROC?
AUROC and ROC, sir?
Yeah, AUC ROC, you have told, no?
Yes, sir. Yes, sir. Yes,
what
sir.
is-
ROC is actually receiver operative curve. That it's actually a
Hmm.
statistical test which is used to test the sensitivity and specificity of different diagnostic tests. And AUROC is the area under the ROC curve. That is, which is being used to determine the sensitivity and specificity levels of the diagnostic test.
Okay.
Gayatri, can you tell us which disease patient used during in your studies?
Sir, actually, in our hospital, we focused our study on medical intensive care unit and the coronary care unit. So basically, most of the death patients, I'm sorry, you're asking about the death patients, sir, right? The patients who died, the common illnesses. Sir?
Any specific disease?
I couldn't say a specific disease that was identified in the critically ill patients, because most of the patients who died, that 15 patients, had a combination of illnesses, and the mostly identified was the cirrhosis, sir, the liver cirrhosis. And next, after that, we had one of the case with a septic shock following the cirrhosis. So I would say the most prominent, amongst those patients, the one common illness would be the liver cirrhosis, that is the damage to the liver, sir.
Okay.
Thank you, Gayatri. Now you can stop sharing.
Yes, sir. Yes, sir. Thank you, sir.
Thank you. All the best. So moving upon our next presenter, I think she's Soundarya from Alva Pharmacy College. Soundarya, please unmute yourself and start your video and presentation.
Good afternoon, sir.
Yeah, good afternoon.
Sir, can I also share my poster?
No,
If
we are
possible.
not. We can't share your poster right now.
Okay, sir.
You please continue with your abstract. We can understand.
Okay, sir. So, my topic is vaccine production. Vaccines are biological preparations that provide acquired immunity against a particular infectious disease. These can be bacterial or viral, and these also may be live but attenuated or may be killed. The method of vaccine production includes seed selection. In this, a small amount of seed is taken and purified, and records are maintained. And then we grow the microorganisms In the case of bacteria, batch culture and continuous culture are used. And in case of virus, cell culture and bird embryo are used. And then we isolate and purify the microorganism. When we isolate the compound which are different from the desired product are removed, and when we purify, the microorganisms are selectively separated and retain the desired product at highest purity level. And then we inactivate the virus so that the disease is not caused. In the case of virus, solvent detergent system are used because these may be lipid coated. So to destroy their lipid coating, solvent detergent inactivation are used. And in case of bacteria, inclusion bodies, membrane extraction, and capsule extraction are used. And at last, we do quality control test of this. Uh,
Okay, Sundara, you please continue. Please conclude yourself.
At last, I want to conclude that vaccines are vital for patient and safe from many infectious disease, from virus and bacteria. And when ready, undergoes many tests and clinical trials to ensure its safety. Thank you.
Thank you, Beta. Thank you, Soundarya. May we have the questions from the panelists to be asked from Soundarya?
Hello. Hello.
Yeah, Gaurav sir, you are audible.
Soundarya, please give an example of a live attenuated vaccine.
Sorry,
No.
sir. I don't know.
Okay, okay, Soundarya. Thank you. We have one request from one of our presentee, which have earlier didn't present it. I think Nimisha Ravinath, please be ready.
Yes, sir. Okay, sir. I'm here.
Yeah, Nimisha, first start your video.
Okay, sir.
I'm searching your extract. I think this one.
Sir, am I visible now?
Yeah, you are visible. I think your topic is practice of intravenous to oral antibiotic conversion and its influence on patient care and clinical outcome at a tertiary care hospital. Is this?
Yes. Yes, sir. Yes, sir. Correct.
Okay, Nimisha, you please start your presentation.
So shall I start, sir? Okay. So good
Yeah.
afternoon, one and all. I'm Nimisha from Nehru College of Pharmacy, Department of Pharmacy Practice. My project is practice of intravenous to oral antibiotic conversion and its influence on patient care and clinical outcome at a tertiary care hospital. So we all know that the antibiotic therapy is crucial in effective management of infectious diseases, and its irrational use is major risk factor for the development of drug-resistant organisms. So the early switch from intravenous to oral could be one of the factors that influence the length of hospital stay. So our study mainly aiming that evaluating the practice of IV to oral antibiotic conversion and its impact on patient care and clinical outcome in a tertiary care hospital. So it is a prospective observational study, period of 6 months, and patient demographic details, medical histories, diagnosis were taken, and we calculated the duration of IV to oral antibiotic therapy, length of hospital stay, and results reveal that 68 percentage were converted from IV to oral, and 31 percentage were not converted. And types of conversion were done and among 187 converted cases, and mostly step down cases were seen. And after that, followed by switch therapy and sequential therapy. And there is a statistically significant differences in the rates of conversion of IV to oral therapy of antibiotics with respect to the various types of IV to oral conversion. The p-value, which is considered as less than 0.001 as a significant value, and length of hospital stay and day of therapy were calculated for converted and non-converted cases, in which LOHS was 62% and total day of therapy was found as 68%, And from non-converted cases, total LOHS was 37% and DOT was found to be 31%. So we can see that for converted cases, the days of therapy, length of hospital stay, which was decreased than that of the non-converted cases. So the estimated cost was shown with the increased for non-converted therapy and for converted, which was decreased. So there is a significant difference in cost between converted and non-converted group of patients. And the practice of IV to oral conversion can shorten the duration of IV therapy and reduce the treatment cost without any negative influence on the outcome of treatment. Then rational use of antibiotic may make better for the healthy society, and that will influence in the outcome of the patient in a fruitful way. So the large array of antibiotic medications are suitable for the conversion, and there are various guidelines available in this regard. Each hospital should implement such a guideline. Okay. So in our study, we found that the most of the conversion was according to the guideline. So it's the responsibility of a clinical pharmacist. So all we know that the thoroughly review the medical records and identify the patients who are eligible for the conversion. So in our study, mainly we got mean days for therapy, length of hospital stay, duration of IV therapy were increased for the non-converted groups than the converted groups. So in our study, we found the treatment cost also could be reduced by increasing the conversion, and thus expenditure of the treatment could be minimized. Setting of our study represent daily medical practice on hospital wards without direct intervention, thus making it possible to generalize the results. And in this Indian scenario, we have here to accomplish an ideal IV to oral conversion program in our day-to-day clinical practice. So that's all,
Okay.
sir.
Okay. Thank you, Nimisha. Anybody,
Thank
any
you, sir.
questions from the panelists to be asked by Nimisha? Any questions from the panelists? Rajesh sir, Mujib sir, Nitin sir, Gaurav sir.
Nimisha, can you tell us these self-medications which taken by the patients, the antibiotic is good to taken as a self-medication?
No,
In
sir.
initial stage.
Antibiotics is not such a good medication for all the patients because it will produces more of the resistant organisms. So in the world, most of the it's a chocolate of prescription, right? Antibiotics is a chocolate of prescription. So it cannot be used for all the patients in so many days. So it will cause most of the resistance in the patients. So it's not good medication for the self.
Actually, most of the practitioners they prescribe the antibiotic initially. So, as a
Yes,
pharmacist,
sir.
how you can suggest them?
Yes, sir. So after antibiotic susceptibility test, we could go for an antibiotic therapy. So by the antibiotic susceptibility test, which will be giving more information on what type of drug can be given to the patient and what type of organisms is associated with the patient. So according to that, we could prescribe such a antibiotic for the patient. So that will be good for the patient. Also, it will be a suitable regimen for the patient.
Okay, thank you.
Hello.
Oh, okay, thank you, Nimisha. Next,
Thanks, sir.
next participant, please be ready. Parul. Is Parul is there? Parul. Anybody left from the selected participants? Those who got mail for poster presentation or oral presentation, who want to present here right now? Actually, we are going to end this session because we have quiz also remaining. So, if anybody from the participants is anybody remaining for oral and poster presentation for those we have selected?
Sir, I'm Kyle Vidi.
Yeah.
Sir, sorry for
And
delay, sir. Due to some network issues, I can't join at the time, sir.
okay, have your poster is selected? Have you received the mail for correction of poster?
I think I got the link, sir, Zoom link.
I think she is selected in oral presentation.
Okay.
As Kyle Vizina.
Yes, sir, Kyle will say Ladan.
Kyle, please start your video first.
Yes, sir. Can I visible,
Yeah.
sir?
Yeah, you are visible.
Sir, can I proceed, sir?
Yeah, Mujib sir, you please start.
Actually, she will give the presentation on the phytochemical analysis and pharmacognosed IC of Feverio and butterware plants for the treatment of the migraine. So,
hello, sir.
Yeah, are you hearing me? Hello.
Yes,
I'm
hello,
audible.
sir. I can't hear. Hi, sir.
You can start your presentation, please.
So, can I share my slides, sir?
No, Kyle, maybe right now we are unable to share your slides. You please explain with this abstract.
Okay, sir. I'll see my presentation and I'll be explaining you, sir.
Okay.
Can I proceed, sir?
Yeah, you please proceed. You please start.
Okay. Warm and pleasant good afternoon to dignitaries present over there. I'm Kyle Viri. I'm studying my B.Pharm final year in Dr. MJR Educational Research Institute. And I would like to present my small research work which I have done in my laboratory. That is nothing but we all may be knowing that recently the researchers from California, what they have suggested in the sense in the journal of a pre-reviewed journal MS System in on 18th October 2016 they have been published that one of the reason for causing a migraine is a gut bacteria. This gut bacteria may be the one of the reason for causing migraine. They have been suggested that what is my solution for this particular migraine disease in the sense basically we may be thinking that how the migraine will how the gut bacteria will cause the migraine. General pathophysiology we may also knowing that once this gut bacteria may be coming from the sources from leafy vegetables and some wines which will be our daily diet and as well as daily food what we are taking. So what pathophysiology involved in the sense once we assume this particular vegetables as well as some wines, this nitrates in the particular food may be converted into nitric oxide. This nitric oxide may be entering into the blood vessels and it will be causing a dilation in the blood vessels which leads to the causing the disease called migraine. So what is my solution in the sense? I have been taking two plants that is nothing but feverfew as well as butterbag. Why I have chosen this particular two plants in the sense these two plants have a very good migraine effect once which I have been clarified using some review as well as research articles, and I have been extracted this two plant and I have taken the extract and I've gone through biological studies that is nothing but antibacterial studies as well as antifungal studies. In the both the studies I have got a positive result when compared to the standard antibacterial drugs as well as antifungal drugs. So what the research says in the sense the ethanolic and I have been done many extract like aqueous extract and ethanolic and many solvent extract, but only in the ethanolic extract I have got some result when compared to the standard. So what I concluded in the sense since I have got a very good antifungal as well as antibacterial effect, so this can be these two plants can be used in a market for the further new drug development for the treatment of migraine. And I have been with this positive result and I have been approached for a research grant in my particular university. So once this grant has been sanctioned, I'll be proceeding with my planned work, sir. That is nothing but I would like to prepare a probiotic mouthwash which can be alter the balance of bacteria to help the prevent and migraine, sir. So this is my small work which I have done in my laboratory, sir. So thank you all, sir. Yes,
Hello.
sir.
Yeah, can you please tell me the active constituent of your plant which are responsible for treatment of migraine?
Pathoniloid, sir. This is a particular
Is it an alkaloid or anything else?
alkaloid, sir. Yes, sir.
Alkaloid. Okay.
Yeah. This is my small work, sir.
Okay, now work is small. You are doing your job very well. Yes, Mujib sir.
In your title, you shows you the biological activities
Sir, I can't hear you, sir.
anti-migraine. Hello. In your title, you shows the biological activities migraine. I think, okay, anti-migraine. But in the abstract, so many activities showing antihistaminic, anti-leukotriene activity. So
Yes, sir.
all you have done these activities.
No, sir. Only anti-migraine activity I've gone through, sir. But fever as well as buttermilk, I have all the activities, sir. What I have included in the abstract, sir. This is a general activity which has that the particular plant, sir.
Okay. So which part you have used during the extraction?
Leaves, sir. Both the plant I have used the dried leaves, sir.
Dry leaves.
Yeah.
Okay. So which solvent is best during the extraction?
Ethanolic solvent, sir.
Ethanolic.
Yeah.
You doesn't use the water as a
Sir,
extraction.
I have used 3 types of solvents, sir. Hexane, ethanolic as well as aqueous, sir.
Aqueous. So
Yeah.
the activity, which solvent gives the best activity during your
Ethanolic, sir. I have
ethanolic?
concluded ethanolic, sir, is best.
Actually, sometime it gives the antimicrobial activity, ethanol itself.
Yeah.
So if you take the aqueous extract, it's maybe more beneficial than the ethanol, I think.
Okay, sir.
Okay, sir.
Okay, thank you participants, thank you presenters for being with us for oral and poster presentation. We are going to end this session and after 3 p.m. We again move upon our valedictory ceremony. So please be with us at 3 p.m. with the same webinar link, not with this link. Earlier link which you have joined with webinar. The valedictory ceremony is going on the same that link. So track 3 frame, which is valedictory ceremony, is on 3 p.m. today itself, and prize announcement is there. So all the best, all the participants. I think if Jayapalan sir is here, I would like to, I request him to ask few words for the participants. I think Balan sir is not here. Mujib sir?
Yes, sir.
Okay. Shall we end this session, Mujib sir?
Yeah, thank you to all for your wonderful presentation. And I wish all the participants all the success. Thank you to all.
Thank you, everyone.
Hello, sir.
Yes, yes.
Sir, actually, we didn't get that initial link. We have only Google Form link and this invitation link.
Then you will receive the link in a few minutes
Okay, okay.
for joining the webinar at
Okay.
3 p.m.
Okay, sir. Okay, sir. Thank you.
Okay, please
Okay, sir.
stay tuned.
Okay,
Okay,
sir.
thank you. Mujib sir, first I will get confirmed from Balan sir for the proceeding. Okay.
Yeah.
हाँ मुजीब सर, आगे क्या है?
गुरप्रीत सर आ रहे हैं अभी।
अच्छा अच्छा अच्छा, ठीक है सर।